Thank you all for coming. We do have lunch, amazingly. So take it, stash it, eat a little bit each day. But it's my great honor to introduce Dr. Peter Hunt, who's joining us today from San Francisco. When I woke up this morning and saw the dense fog, I thought, oh man, it comes all the way down here to LA and it's foggy, but finally the sun has come out, so he gets to see why it's so great to be in Southern California. Peter's a professor of medicine in the Division of Experimental Medicine, one of the co-directors of the UCSF CFAR, and also a scientific director for the AIDS Clinical ACTG Laboratory Center, and has really had a tremendous career doing translational research, and one of the people who really has pushed the envelope around persistent inflammation in people living with HIV. He's done some great work in mucosal immunology, and also throughout his career has had this interest in CMV and how in its role in driving some of this chronic inflammation. So I'm going to let him tell you about this really exciting story that's developing around CMV inflammation and aging in HIV. Welcome, Peter.
Thanks. Thanks, Judy, and thanks for asking the sun to come out. Really appreciate it. So let's see. That button didn't work. Let me try this. All right, here are my disclosures. To set the stage, the life expectancy gap for people living with HIV in the modern treatment era has been narrowing compared to the general population. You can see here people with HIV. These are data from the Kaiser cohort. They're about a decade old now, showing that about a 9-year gap in life expectancy compared to people without HIV. And if you start ART at a high T cell count, that gap narrowed to about 6 years. Still, we've made a lot of progress, but still a gap. And that gap is actually greater for women than it is for men. These are more recent data from the ART-CC collaboration. Over 200,000 people with HIV around the world, tracking life expectancy versus the population average in people without HIV. And that gap, if you started at a high CD4 count, is narrowed to 1 year in the lower risk categories of men with HIV. And if you started at low T cell counts, there's still that persistent gap. But if you look at women, there's a much greater gap. So down at the lower naders, 24-year or 14-year gap rather in life expectancy. And even in those who start at high T cell counts, there's a 3-year gap in life expectancy. Now, they're living, around the same life expectancy as men with HIV. It's just that in the general population, women live longer, and that there's a loss of female advantage in life expectancy with HIV. And a lot of people have thought about the behavioral risk factors that might explain this. Even when you control for those factors, you tend to see these differences. And I'll point you to Rebecca Abelman's paper that's in press now at JCI. You should invite her to a Grand Rounds soon in the next year, and she can tell you about her story that might explain this loss of female advantage in HIV. But I'm going to come back to sex differences at the end of this talk as well. It's also true that people with HIV are accumulating more and more comorbidities associated with the aging process. Heart disease, cancer, liver disease, renal disease, osteoporosis, et cetera. And on average, are getting those conditions about 15 years earlier. And even people who start at high T cell counts, it's about 10 years earlier than the general population. And that gap is not narrowing. So we're still living with multimorbidity, and those of you who do HIV clinics see this all the time. Priscilla is here. She sees a lot of your patients with heart disease. There's an increased risk of multimorbidity in the context of HIV. And here's just a list of many of those conditions. I'm highlighting a few that I think are particularly important and underappreciated by our HIV treating a community. Infection-related cancers, extraordinarily high in people with HIV, much higher than in the general population. Also lung cancer and other hot tumors where checkpoint inhibitor therapy has become the mainstay. Those types of cancers are increased in people with HIV. And that is something that is underappreciated by a lot of people. Also frailty, declining functional status that we normally think about in much older populations you see, particularly in people with HIV with low CD4 nadirs. And so what might explain a lot of this? Well, this is my favorite medical journal, Time Magazine, back when I was, around the time I was starting my research career in the early 2000s, already in the popular press, this idea that inflammation might be contributing to chronic diseases of aging was already gaining popularity. At the time, we didn't have really specific therapies to go after this, but at least this was an idea that was circulating even in the popular press. And we were studying back then these markers of T cell activation, which Janice Georgie had really put forward for right here at UCLA in the pre-treatment era, and then early during ART. We took some of those same markers, 38DR expression, and saw that compared to untreated people in red, people with treated HIV with suppressed viral loads in green had lower levels of immune activation, abnormally high compared to people without HIV. And we came back, and these are data that we repeated several years later with a larger sample size and with a more robust HIV negative control group to confirm these associations. And several other groups later showed that markers of innate immune activation and inflammation also remained abnormal in people with treated HIV and predicted subsequent disease risk. Most famously in the SMART study, and then these are data from the Insight Network combining across the control groups of many of their treatment strategy trials of ART-suppressed people followed for 10 years. Single measurement of IL-6 dramatically predicts the risk of morbidity and mortality. Over 20% chance of one of those significant events over 10 years in people in the top quartile compared to only about 5% in the bottom two quartiles. And the curves are continuing to separate over time. And that's always suggested to us that there's likely to be an inflammatory set point within individuals, some people who have high inflammation, who are at high risk of disease, and some people with low inflammation who probably don't need new interventions. And so our field's goal is to safely move people in the top two quartiles down to the bottom two. That's what we want to do. I also want to highlight this link between inflammation and frailty and declining functional status. In the geriatrics literature, this has been well known for a long time, that inflammation may not only induce muscle atrophy, but may also suppress muscle repair and regeneration programs, all of which leads to muscle atrophy. So think of that hospitalized patient who's been in bed for a week, but also hospitalized with an inflammatory condition. Those two things conspire to result in quite a lot of muscle wasting. And we see this at a much lower level, but a clinically significant level in many people with HIV. And so what do we know about the causal relationship between inflammation and disease outcome? We don't know a lot yet in the context of HIV, but we know a lot now in the general population. These are data from Paul Ridker's group, also collaborator of Priscilla's, cardiologist in Boston, and has been at this idea for a long time that inflammation is causally associated with heart disease. First looked at statins. People said, well, statins affect LDL. He said, okay, I'm going to come at you with an IL-1 beta inhibitor, part of the NLRP3 inflammasome, a root driver of inflammatory processes. And so IL-1 beta blockade with canakinumab resulted in a 15% decline in cardiovascular events. First proof that inflammation is causally associated with heart disease. Even more compellingly, so he's a cardiologist, so he did the study for heart attacks, but even more compellingly, there was a 50% reduction in cancer mortality. 50%. And 75%, 77% reduction in lung cancer mortality. There was also a decrease in lung cancer incidence in people who got canakinumab. And that just blew my mind. So a hot tumor, remember that. That inflammation seems to reduce those comorbidities even more than heart attacks. This, again, is the first proof that inflammation causes disease. Of course, all of our patients are not getting canakinumab for this indication because there was a competing risk of increased death from sepsis. And so it was not FDA approved for this indication, but it was proof that inflammation could drive some of these comorbidities. So what have we done in the HIV field? So I call this treating immune activation version 1.0, where we test commonly used safe medications with anti-inflammatory properties. In the ACTG, we've studied with Judy and many of these interventions. We studied aspirin, there have been ARBs, ACE inhibitors, a whole host of interventions that have anti-inflammatory activity. Most of them failed to reduce immune activation, or at least the markers we care about, except statins showed some benefit in early studies. And so that led to the famous REPRIEVE trial, which many of you are familiar with. This changed clinical practice. Now most of our patients are on statins to reduce heart disease risk, because even in people who didn't otherwise have an indication for a statin, pitavastatin reduced the risk of heart attacks in people with HIV by 35%. Really strong effect. Absolute risk reduction 3.4 to 2.4%, so 1% at 5 years. But when you looked at the major non-cardiovascular events, non-atherosclerotic cardiovascular events, there really wasn't much of a difference. Now, a lot of us were hoping that maybe the pleiotropic effects of statins, the anti-inflammatory, special sauce of statins might end up helping reduce all these other events, cancer and other things. But we really didn't see that. In fact, the two leading causes of death in people with HIV in the United States, it's cancer, 3% in both arms, 3% incidence, and TB. This was an international study where we had a fair amount of TB in sub-Saharan African sites, 1% in both arms, no different. So we weren't affecting the leading causes of death. And if you look at all of these non-atherosclerotic major clinical events, the incidence is much higher than it is for atherosclerotic cardiovascular disease events, 4.9, 4.3% at 5 years. So that's an important point. We're missing the majority of the clinical events that people with HIV have with just statins alone. So we need extra intervention. There was also no effect on congestive heart failure with statins, no effect on frailty, no effect on cognitive function in other studies. And so Peter Rice and I wrote an editorial on this paper earlier this year, pitavastatin not so pleiotropic in HIV. So it's the statins are great at reducing cardiovascular risk, but that's because they reduce LDL and perhaps improved plaque stabilization. You may have seen this paper from the REPRIEVE investigators identifying P-Colcy and some other proteomic markers associated with atherosclerotic plaque stabilization that might be playing a protective role. But they really saw non-significant trends towards a reduction in IL-6, CRP, despite measuring this in nearly 700 people. Just a really, really weak anti-inflammatory effect. And when you look at the correlation between the reduction in non-calcified plaque by CT while you see a strong association for LDL, you see very little correlation with suppression in CRP. So statins work by reducing cholesterol, maybe stabilizing plaque, but it was not an anti-inflammatory effect in HIV. And that may be why we didn't see a reduction in all those other non-atherosclerotic events. So the other approach our field has taken, I call this version 2.0, is to go after the inflammatory pathways themselves, kind of like canakinumab, right? Go after the inflammatory response directly. I do think there's a whack-a-mole problem with this approach. And by that, I mean that you may interfere with immune defenses, increasing infection risk, as was the case in the CANTOS study. You might not block all important inflammatory pathways. I used to use this analogy of a tree and go after the tree trunk. If there's a common inflammatory pathway driven by all the root drivers, maybe it makes sense to go after that. But the problem is there may not be a single tree trunk. There may be multiple parallel inflammatory pathways, and going after this one cytokine might not solve the problem, and it may even make some of the other pathways worse by interfering with some of the root drivers of the inflammatory state. And we've had multiple examples of so-called tree trunk interventions or going after these inflammatory pathways that have shown mixed effects. Some benefits in some pathways, but either infection concerns or worsening risk markers for some other pathways. And so whack-a-mole is a problem with version 2.0. And so version 3.0 is actually where I've always been. I've always felt that we needed to be looking at the root drivers of inflammation rather than the mediators themselves. I focused a lot of my early work on CMV, not so much on HIV and microbial translocation, because we haven't had interventions that block HIV from coming out of cells yet. We haven't had direct and effective and specific interventions to block microbial translocation that work, although we've tried a few that didn't help. And so I've spent a lot of time focusing on CMV. I think we're close to getting interventions for HIV expression from cells, or at least clearing HIV expressing cells. And I think that's going to be an important area of research. But the rest of my talk is going to be about CMV. So why CMV? Well, CMV is induced to replicate by inflammation. So it could be HIV reservoirs, microbial translocation from HIV, inducing TNF alpha locally in tissues, inducing lytic replication of a herpes virus. This happens really with all the herpes viruses. They're induced to replicate by inflammation. And once it's replicating, it can either amplify local inflammation. Now you have a whole other virus around contributing to local inflammation. But CMV is a tricky virus, and it encodes lots of immune evasion strategies, lots of proteins that help it evade the immune system, including its own viral IL-10, which is immunoregulatory. And I'm going to get into that a bit later. It's a very key part of the story that I'll be telling you. But at least going into this, we had a very simple-minded idea that it's just going to amplify inflammation. And so maybe if we dampen down that amplifier, it'll improve immune activation. And we had a lot of support for this concept, working with Mike Freeman at Case Western Reserve. We looked at people who were CMV seropositive versus negative with HIV. The seropositives were the only people where we saw substantial CD8 T cell expansion. Normally, we see that low CD4 to CD8 ratio, expanded CD8s. This is primarily observed in people who are CMV seropositive. Markers of interferon response, IP10, soluble TNF receptor 2, and D-dimer all increased among seropositives versus seronegatives, and a whole host of other biomarkers. Other groups, including Renee Reddy's group, have linked to increased clotting and thromboembolism risk as well. It made us think that we were on the right track in targeting CMV. And here's Priscilla, live and in person in the front row. This is our first study together all those years ago, it identified CMV as potential surrogate marker or potential factor associated with atherosclerosis. And so in red are people with HIV, and blue are people without HIV. X-axis is CMV-specific CD8 T cell frequency, and then you have the extent of atherosclerosis on the y-axis. So all the people with HIV are up here in the upper right-hand corner, and the people without HIV are down here. And actually, in this study, if you adjusted for differences in CMV-specific T cell responses, the increased atherosclerosis in people with HIV went away. And that was an early clue that CMV might be a potential mediator. Much later, in the Italian Icona cohort, they had quite a number of CMV seronegative people with HIV. In San Francisco, it's over 95% of people with HIV have CMV. Hard to do a study. In Italy, they had more CMV seronegatives they could compare. And the CMV seropositives had a higher incidence of non-AIDS events, particularly for cardiovascular events, with a hazard ratio of 2.3, quite a strong effect, suggesting a potential link to heart disease with CMV. In the general population, you don't see that, by the way. In meta-analyses done recently, CMV does not seem to be a major determinant in people who don't have HIV and otherwise don't have an inflammatory state. But there's something about HIV and perhaps having low-level inflammation that where CMV tends to play a more important role. We've also looked at cancer risk with Sam Schmidtman and the two Gabbies in my lab. We looked at CMV IgG titers predicting some of those cancers that are increased in people with HIV, non-Hodgkin lymphoma, anal and lung cancer. And we see really a strong association with a lot of inflammatory markers, but also CMV specific IgG titers. The ACTG several years earlier had done an analysis on all non-AIDS cancer and also saw the same thing. CMV-specific IgG titers predict increased risk. EBV-specific IgG titers do not. So then our first crack at getting at causality came over a decade ago. This is my first clinical trial of valganciclovir in people with treated HIV to see if asymptomatic CMV replication might be contributing to immune activation. And we were using for the primary outcome those old T cell activation markers. My friends and colleagues, Becky Ho, Steve Deeks, helped mentor me on this back in the day. We did this within the SCOPE cohort and embedded this within the SCOPE cohort. And sure enough, valganciclovir reduced T cell activation, an effect that persisted for 4 weeks after we stopped the drug, which is an important observation that I'll come back to in a little bit. But at the time, we didn't really move forward with this because valganciclovir had lots of toxicities, cytopenias in particular, that made it unattractive for a longer-term trial. And ultimately, if we wanted to reduce end organ disease, we have to give people something long-term, and so we needed something safer. So we kind of waited until our colleagues in pharma developed newer drugs. And as we were getting closer to having new drugs available to study, we went back to samples from that early trial of valganciclovir and looked at other plasma markers that were shown to be more strongly associated with subsequent cardiovascular events and cancer, namely soluble TNF receptor 2, soluble CD163, and those significantly declined in the valganciclovir arm. So we again, we thought we were on the right track here. But it was always unclear to us whether this effect was mediated by a reduction in CMV replication versus effects on other herpes viruses. EBV, importantly, but also HHV6, KSHV, HSV1 and 2, VZV, they're all suppressed by valganciclovir. It's really a pan-herpes virus drug. So we really didn't know, which virus was responsible, or a combination of viruses was responsible for this. And to sort of drive this home with CMV and EBV, CMV was suppressed through 8 weeks of treatment. And again, even for 4 weeks after we stopped, we also continued to suppress CMV shedding, which may be responsible for that delayed immunologic effect. Again, I'll come back to that again in our next study. But EBV, you see, clear suppression with valganciclovir. So we are, and we also looked at KSHV. We looked at HHV6. We saw declines, less significant declines, but declines in those viruses too. So then came letermovir, and letermovir is a CMV terminase complex inhibitor. That's pictured in the cartoon here. It's when CMV replicates, you get these long concatenated strands of CMV DNA that are repeated, and the terminase complex clips those DNA genomes and packages them into the capsid. And the key thing here is that the drug is specific only for the CMV terminase enzyme. It's not shared by any other herpes virus, and so it's really one of the only drugs that's specific for just a single herpes virus. So we can isolate the effect of treating CMV. And so working with Sarah Gianella at UCSD, we designed this trial in ACTG, the letermovir trial A5383. The ACTG likes those numbers. I can never remember them. I remember this one though. And we treat people for 48 weeks with letermovir versus no CMV treatment, followed by 12 weeks of observation. We were really curious to see what happens at longer periods of time off of drug, to see if there was continued suppression of shedding. We specifically enriched for people with lower CD4 counts because we thought the drug might be more active in them. More similar to the population we studied with valganciclovir. And we also made sure that a third were women because up to this point, there had been quite a lot of evidence that women respond to viral infections with a more robust inflammatory response than men. And so we reasoned that women might stand to benefit more from this intervention. And so now, we also built in lots of sub-studies to this. Some may say too many sub-studies. And the ACTG required that we build in a futility analysis because we had so many bells and whistles. Make sure that after the first 40 patients reach week 8, that you're on the right track in terms of the immunologic measurements, soluble TNF receptor 2, soluble CD163 before moving forward for the rest of the 180. It's an expensive and long study. And so this happened in the summer of 2023. These are the characteristics of the people in the futility analysis: 18 in letermovir, 21 no CMV treatment. Per design, we had nearly a third women. And also by design, nearly half had CD4s less than 350. Here's the antiviral activity. We did have 2 participants at the week 8 visit that had barely detectable CMV DNA in semen in men. We looked at CMV shedding at multiple sites, oropharyngeal washes, semen in men, cervicovaginal swabs in women, and also rectal swabs. Just 2 samples were positive early, but not at no later time points did we see any more CMV. during the treatment period. So it worked. It had a good antiviral activity, and you can see the shedding rates in the control arm. And importantly, all plasma CMV DNA levels in the entire study were negative. So all of this is happening in the tissues. None of it is happening in the blood. So what did the futility analysis show? The exact opposite of what we predicted. Soluble TNF receptor 2 actually increased and significantly within the letermovir arm, and a trend in the wrong direction for soluble CD163. We did not meet the futility analysis, and the trial stopped early. And as my former mentor and colleague Steve Deeks is fond of saying, this is my specialty, where my a priori hypothesis is not just wrong, it's 100% wrong. But it's also my favorite result in science because you always learn something new and it makes you think about things in a new way. And so in retrospect, the declines that we saw in these two markers in the valganciclovir trial must have reflected effects on other herpes viruses besides CMV. And not only that, at least for soluble TNF receptor 2, those other herpes virus effects dominated over the effect of CMV because treating CMV actually increases soluble TNF receptors. So there was a dominant effect on one of the other, at least one of the other herpes viruses. So then we really got to work thinking, so what could have explained this? And remember, I told you from the beginning that CMV is doing a lot of different things. On the one hand, it may be amplifying inflammation. They're through multiple pattern recognition receptors on bystander dendritic cells. You can get an inflammatory response. You can also, via IL-1 beta, also TNF alpha production and antigen presentation. But I told you that CMV is also making this immunoregulatory viral IL-10. When it binds to the IL-10 receptor, it actually suppresses TNF alpha production by dendritic cells, and it also suppresses antigen presentation. So class 2 HLA expression that goes down. The immune system is not seeing CMV. It's a way of hiding from the immune system. But this is effectively a brake on the immune system. And so CMV kind of has its foot on the accelerator and the brake at the same time. That's what we think was happening. And so when we come in with letermovir, we're blocking both, we're removing our foot from the accelerator and the brake. And so you have this mixed picture. We may have a reduction in inflammation in some pathways because we're blocking that amplification step, but we're also increasing antigen presentation. We're removing that immunoregulatory step and perhaps increasing TNF alpha production, at least early on. And we had evidence that this was happening. We, using the Olink proteomics platform, we measured changes in IL-10 receptor alpha, which is the specific part of the IL-10 receptor, which is shed after IL-10 engagement with the receptor. So can be, we don't know exactly how it ends up in the plasma, but one of the ways is through IL-10 receptor activity, and it did go down in the letermovir arm, and it was significantly different between arms, whereas IL-10 levels didn't change at all. So something was reducing IL-10 receptor activity. We think it was a CMV viral IL-10. We're not able to measure CMV viral IL-10 reliably. There is an ELISA assay that's published. We are not confident that assay is truly specific for CMV viral IL-10. We think it's cross-reacting with another human protein. We haven't figured that out yet, but we don't think we can reliably measure this. Interestingly, we did not see a reduction in IL-10 receptor alpha in the prior valganciclovir trial. So why the heck might that be? Well, so Yoshi Fukuzawa in my lab, we got to work first just trying to confirm some prior published studies showing that CMV's viral IL-10 is actually functions as a superagonist. It actually is more potent at inducing IL-10 receptor activity than human IL-10. So CMV IL-10 is in red, human IL-10 is in blue, and EBV also makes a viral IL-10 in yellow. And you see that in each cell type we tested this in, and monocytes have the highest IL-10 receptor expression. So you see the biggest signal in STAT3 signaling here. You see in every cell type, CMV outperforms human IL-10, and EBV is way down at the bottom. So it's a partial agonist. And if you pump up the dose of the IL-10, EBV can induce signaling, but it's just much, much weaker than CMV and human IL-10. And so you have these two herpes viruses that have qualitatively different viral IL-10s. And I think that's a really important clue. And we started wondering whether CMV might be immunoregulatory and EBV more pro-inflammatory on this basis. That EBV's viral IL-10 is a partial agonist, CMV's is a superagonist. And it turns out that polymorphisms associated with increased IL-10 receptor signaling are associated with an increased risk of CMV retinitis in people with AIDS. So potentially linking IL-10, the IL-10 pathway to pathogenesis of CMV. Polymorphisms associated with decreased IL-10 expression are associated with an increased risk of multiple sclerosis. And you all know that EBV has famously been linked to an increased risk of MS. And I'll talk about that in the next slide. But so why did valganciclovir not change IL-10 receptor activity? Well, we think we removed a superagonist and a partial agonist at the same time, and that the two effects likely canceled each other out. And there's far more EBV virus made in all the mucosal sites we tested than CMV. And we think there may be an immunologic mechanism for that. This very same mechanism may suppress CMV replication while amplifying EBV replication. So we think that's the reason we didn't see anything in the valganciclovir trial. And these viruses have qualitatively different effects on other disease associations. So everyone knows this famous science paper, EBV, incident EBV associated with dramatically increased risk of multiple sclerosis. What few people remember from this paper, including some of its co-authors, CMV seropositivity is associated with a decreased risk. Now, the y-axis is such that you can't really appreciate the effect size so much, but this is a significant decreased association. And a year earlier, or a year earlier, in Europe, an independent study was done seeing the same thing. EBV and HHV6 infection were associated with an increased risk of multiple sclerosis, but CMV associated with a decreased odds of 30% decreased odds of multiple sclerosis. We saw the same pattern in long COVID. So working with Michael Peluso in the LINK cohort at UCSF, we saw that while, as others did, that while EBV reactivation and then also HIV infection was associated with an increased risk of neurologic long COVID symptoms, CMV seropositivity was associated with a 50% decreased risk of long COVID. So exact same pattern of associations. And so consistent with this idea that maybe CMV may be playing an immunoregulatory role here. So we wondered whether those early letermovir-mediated increases in immune activation may have been due to a loss of the immunoregulatory viral IL-10. But what happened with longer-term letermovir treatment? So the ACGG thankfully let us continue to follow people during the study, and for a variety of complicated reasons, it took a long time to finalize the data for the futility analysis. And so most people actually made it all the way to the end of treatment before the trial was stopped. And when we followed people, this is what happened. While it did go up early, soluble TNF receptor, it ended up going down at the end. And significant and highly significant difference in both of these segments and between arms. And we saw similar patterns for other plasma cytokines, CRP, IL-6, and the coagulation marker D-dimer. When we looked at earlier, when we looked at other cytokines that we know are causally linked to heart disease and cancer, like IL-1 beta itself, it had a different pattern. It declined early and stayed down. And that we got really excited about, given the known causal links between this pathway and disease. We then looked at O-link proteomics, the inflammation and cardiometabolic explorer panels, so 750 or so proteins. And you can see from across the room that this volcano plot is sort of tilted to the left, meaning that more things declined with letermovir than increased. And you can see the types of proteins that decline, some inflammatory proteins, a lot of complement pathway proteins, endothelial dysfunction proteins, angiogenesis proteins that are linked to cancer pathogenesis also declined. And so we were really excited about these results as well. And then we decided to hone in on things so we had the IL-1 beta data from clinical trials. But the Insight Network had done the first Mendelian randomization study in HIV, linking, leveraging the power of host genetics for causality of these proteomic associations. So not just does the protein predict disease, but does the genetic polymorphism that changes protein levels also contribute to disease? That's strong evidence of a causal relationship. They identified these 5. 3 out of the 5 declined with letermovir. And so we started to think that, gosh, we had this, counterintuitive result and disappointing result to our team initially, but these data started making us think that we were actually on the right track, that we chose the wrong primary endpoint, but the overall idea was probably right. Interestingly, there's no evidence for causality for soluble TNF receptor 2 or IL-10. And this is also confirmed in recent study in the at least your soluble TNF receptor 2 in the UK Biobank. It's strongly associated with an increased risk of subsequent cardiovascular events, but the genetic determinants of it are not. And so it's not a causal relationship. So if we knew that when we were planning the clinical trial, we wouldn't have chosen this as an outcome, but now we know. So other markers associated with immunosenescence in elderly populations, like the CD4 to CD8 ratio, they increased significantly in the letermovir arm. CD4 to CD8 ratio has also been linked to cancer risk, in particular infection-related cancer risk in people with HIV. So this effect size over just 1 year corresponds to about a 14% reduction in non-Hodgkin lymphoma and anal cancer, some of the more common cancers that we see in people with HIV. And then the most exciting thing to us was this. I told you we were interested in frailty. Christine Erlandson really helped advocate early on to include these markers of physical function in the trial because of that. And I really thank her for that. And the key measure here is the 5-time chair rise time. So if you stand up 5 times from a sitting position without using your hands, and do it as fast as you can, that's a really good measure of your leg strength and physical function. And it's used in a lot of geriatric studies. So we did this in the trial. Yeah, I tried, everybody. I'll time you. it improved by nearly 2 seconds in the letermovir arm. And so when I first saw this, like, okay, it's one measure, and I'll tell you that all the other frailty measures that we measured were not significant, but they all trended in the same direction. And Christine tells me there's a lot more noise in a lot of those other measures. But we were still a little bit, anxious. This is maybe just a fluke. But then we saw that it was strongly correlated with the extent of immunologic improvement. So those with the greatest improvement in CD4 to CD8 ratio had the greatest improvement in chair rise time. We saw the exact same thing for changes in IL-6 receptor and IL-1 beta. People had the most reduction in inflammation, had the most improvement in the chair rise time. And that got me really excited and that was true even within the letermovir arm itself. So that got me really excited that we were on the right track. So I'm going to, the last data slides I'll show relate to interactions. I told you we thought it might work better in women than men. Here's a volcano plot where to the left means that things decline to a greater degree in women than in men. And to the left means they decline to a greater degree in men. And you see there's more stuff that declines in women. And a lot of these are statistically significant interactions. And many of them are involved if you do like a KEGG pathway analysis involved in heart disease risk and cancer risk pathways. We also saw interaction by CD4 count. Now here, a greater decline in people with lower CD4 counts is to the right. And you see that there are more hits there too. So we're also seeing a signal that it tends to work better than in people with lower CD4 counts, at least for the proteins listed here. Though lots of inflammatory markers like IL-1 beta still decline in people with high T cells. But we were starting to think about this sex interactions. So we saw the volcano plot, and then we were thinking about those early changes that we saw, and we started doing a little bit of a literature search. And just a couple of years ago, a group in Vancouver had reported that cells from females have impaired or decreased IL-10 receptor signaling than cells from men. Same amount of IL-10 receptor on the cell surface, same amount of IL-10 being challenged, less STAT3 signaling in women, resulting in a more robust inflammatory response to LPS, because that normal break of IL-10 receptor signaling is less strong in women than men. Now, this is just one study, and we're actually actively working on trying to confirm this and then really dissect out all the age, and hormonal contributions to this. But that got us excited to look at what was happening early with the soluble TNF receptors. And this is what it is, stratified by sex. In men, we see that pattern. Most of the people in the trial were men, and we see that striking increase in soluble TNF receptor followed by a decline. It went down early in women. We didn't see that. Remember, CMV is doing two things: immunoregulation, but also pro-inflammation. And so in the women, the foot was not on the brake in the beginning. They weren't responding, we think, to the IL-10. And so the only part that we saw was a reduction in inflammation early. Had we done the trial only in women, we never would have stopped. Amazing. Now, this fits with this hypothesis that there's a difference in IL-10 receptor signaling. There are issues with that paper that were published, and so we're trying to do a more rigorous analysis of this to confirm it. But there's something very interesting that happened here. I'll also say that there was also no change in IL-10 receptor alpha levels in women. It stayed flat. So consistent with this idea. All right, so I'm going to stop there and then take questions. So treating asymptomatic CMV with letermovir proves immunologic and functional aging in treated HIV, at least in this pilot study. There is a transient increase in some inflammatory markers, which we think may be due to suppression of the immunoregulatory CMV viral IL-10. There's an early and sustained decrease in other inflammatory pathways that increase cardiovascular disease and cancer risk, namely IL-1 beta. It's also improvement in the CD4 to CD8 ratio, improved physical function, which correlated with that improvement, better improvement in women and in those with lower CD4 counts, that transient early increase in inflammation was not observed in the valganciclovir trial, we think, because we were suppressing other herpes viruses too, in particular EBV, which has that partial agonist. And while the clinical implications remain unclear, we're actively planning our next study, and we think these findings may have broader implications, not just to HIV, but also the aging field, geroscience, and transplant fields. With that, I'll stop. I want to particularly acknowledge Sarah Gianella, my co-chair for the letermovir trial, standing in front of our initial poster on the trial, and all the co-investigators and funders, including the ACTG. So thanks, and I'll take your questions.